Let's put the whole question up first, options and all. An investigator measures how fast glucose gets into isolated fat cells, adipocytes. They use two sugars, D-glucose, the normal one your body runs on, and L-glucose, its mirror image, each radio-labeled so its entry can be tracked. 3 results stand out. 1. At every concentration, D-glucose enters much faster than L-glucose. 2. As you add more D-glucose, uptake speeds up, then flattens to a plateau. It does not rise forever. 3. Adding insulin pushes that plateau higher, and the whole process burns no ATP and never moves glucose uphill against its gradient. So the question is, which mechanism best describes how glucose gets into these cells? A. Simple diffusion. B. Receptor-mediated endocytosis. C. Facilitated diffusion. D. Primary active transport. E. Sodium glucose cotransport.
Right, the whole experiment is on the table, and I have to commit to just one mechanism.
Exactly. Pause here and say your answer out loud before we start. Choosing first is the part that actually builds the memory. Then we'll take every clue apart and land on the one option that survives. Before any option, one habit. The trap here is to answer from it's glucose, and glucose has transporters, so carrier. You might get lucky, but you didn't read the question. Every single line in that stem is a transport property, put there on purpose, and each one rules something in or out. We're going to earn the answer off the experiment.
Okay, so the 4 things the stem is actually telling me are D beats L, it plateaus, no ATP, and never uphill.
Right. Hold those 4. 2 of them prove there's a protein at all. The other 2 decide which kind of protein. Let's take them in order.
First clue, D-glucose enters faster than L-glucose. Why does that decide anything?
Because D and L-glucose are mirror images, enantiomers. Same molecular formula, same atoms, the same bonds connecting them. The only difference is their 3-dimensional handedness, which way the groups point in space.
Wait, I want to be careful with the word here. When we say the carrier is specific, isn't that just that it picks glucose over, say, fructose?
Good, that's the distinction to nail. Telling glucose from fructose is structural specificity. Those are different molecules, built differently. What the stem shows is stronger. It's stereoselectivity. Stereo means spatial, 3-dimensional. The carrier is telling apart 2 molecules that are structurally identical and differ only in how they're arranged in space, like a left and a right hand. That is a much sharper claim.
And why is that sharper claim the one that proves a protein?
Because a plain lipid membrane, just oily fat, cannot tell a left hand from a right hand. Dissolving through fat depends on how greasy and how big a molecule is. And D and L-glucose are identically greasy and identically sized. If crossing were just dissolving through, D and L would go at the exact same speed. The moment one is faster, something with a shaped binding site is choosing between them.
Like a glove.
Exactly like a glove. A right glove fits a right hand and refuses the left, even though both hands have all the same parts. That glove is a protein, a transporter, with a pocket built to grip D-glucose and turn its mirror image away. So clue 1, all by itself, says carrier-mediated. Stereoselectivity means a protein is doing the work.
But physically, how does one little pocket tell 2 mirror images apart?
The clean textbook picture is 3-point attachment. The binding pocket grips the sugar at 3 points at once. For D-glucose, all 3 contacts line up and it locks in. Flip to the mirror image, L-glucose, and at most, 2 of the 3 can match. The 3rd contact is now on the wrong side of the molecule and can't reach. 2 out of 3 is a weak, brief grip, so L-glucose barely gets carried. That's why the gap between the 2 rates is so large, not just a little.
Second clue, the uptake climbs, then plateaus. What does the plateau add?
The same verdict, carrier, but from a completely independent angle, which is why it's worth its own beat. Think about what should happen if glucose just diffused through freely. More sugar outside would always push proportionally more inside. Double the outside, double the rate, forever. A straight line, no ceiling.
And instead it flattens.
It flattens. That plateau has a name, the maximal rate, Vmax. A ceiling means there's a fixed number of carriers. Each one can only cycle so fast. Once every carrier is working flat out, fully saturated, throwing more sugar at them can't speed things up, because there's no free carrier to grab it. Saturability is the fingerprint of a finite number of transporters. You literally can't saturate something that isn't there, which is why simple diffusion never plateaus.
So stereoselectivity and saturation both point at a carrier from two different directions.
Two independent proofs, same conclusion. That's about as solid as a question gives you. Now the last two lines of the stem do the rest of the sorting.
Before we sort, can we lay out the whole field once? You had a mental model earlier. Simple diffusion, carrier transport, active transport. Where does glucose really live?
Let's draw the real map because it makes every wrong answer fall out on its own. For glucose specifically, forget simple diffusion. Glucose is polar. It does not dissolve through the oily membrane in any meaningful amount. Glucose crosses on proteins, and there are two families. Family 1, the GLUT proteins, glucose transporters, GLUT1 through GLUT4 and beyond. These are facilitated diffusion. They open a gated channel-like path and let glucose slide down its gradient from where it's crowded to where it's sparse. No energy, no sodium. Downhill only. Picture a revolving door. It only turns the way the crowd is already pushing.
And each GLUT sits somewhere different?
Roughly, GLUT1 is the basic everywhere transporter, red blood cells, the blood-brain barrier, always on baseline uptake. GLUT2 is the liver and the pancreatic beta cell, a low-affinity sensor that tracks how high blood sugar is. GLUT3 is neurons, high affinity because the brain wants first pick. And GLUT4, the star of this question, is fat and skeletal muscle. And it's the one that answers to insulin. Family 2, the SGLT proteins, sodium glucose cotransporters. These are secondary active transport. They drag glucose uphill against its gradient by letting sodium flow downhill at the same time and stealing that energy. Same revolving door, but now sodium rushing in is what spins it, so glucose can be pushed the wrong way.
And SGLT lives where?
Exactly the two places your body must claw glucose back against a gradient. SGLT1 in the small intestine, pulling glucose out of your food across the gut lining, and SGLT2 in the kidney's proximal tubule, reabsorbing the bulk of the glucose you filter so you don't lose it in urine. That's the target the Flozin diabetes drugs block on purpose. Uphill work, so it has to be powered.
So the active transport in the intestine I was half remembering is SGLT1.
That's it. Your instinct wasn't wrong, it just needed names. Two families, GLUT, downhill and free, SGLT, uphill and powered by sodium. Now watch how fast the stem sorts them. The stem's third line, no ATP, and never against the gradient. Two facts, and they knock out two different things. Don't blur them together.
Which one kills which?
No ATP kills the pump, primary active transport, because the pump directly hydrolyzes ATP to force its cargo uphill. Never against the gradient, only downhill, kills SGLT cotransport, because SGLT's whole job is to move glucose uphill. So after this one line, we're left with a carrier that moves glucose down its gradient, spending no energy of its own. On the map, that is exactly one family. GLUT, facilitated diffusion.
And the insulin line, where does that fit?
That's the fingerprint of which GLUT. In fat and muscle, the insulin-responsive one is glucose transporter type 4, GLUT4. Here's the mechanism. Most GLUT4 sits parked inside the cell in little vesicles. Insulin signals the cell to move those vesicles to the surface and fuse them in, so suddenly there are more transporters in the membrane. More carriers means a higher ceiling, the Vmax goes up. That's precisely insulin raises the plateau. Not more affinity, more doors.
Nice, so insulin doesn't change how each transport works, it changes how many there are.
Right, and that's a favorite exam point. Insulin raises Vmax by translocation. It doesn't change the affinity. One more pearl while we're here. Muscle can also recruit GLUT4 during exercise without insulin through contraction itself. That's a big reason exercise lowers blood sugar even when insulin is low. Same transporter, second doorbell. So the answer is C, facilitated diffusion.
Let me say the whole chain back, because this is the part I want to be able to reproduce. Stereoselective D beats L means a carrier. Saturable, the plateau, means a finite carrier. No ATP means it's not a pump. Never uphill means it's not cotransport. What's left is a carrier moving glucose downhill for free, facilitated diffusion, and insulin raising the ceiling names the specific protein, GLUT4.
Perfect. And notice the move you just made. You never once argued from what glucose is. You let the transport properties classify the mechanism. Stereoselectivity, saturability, energy use, gradient direction. Those 4 questions sort every membrane transport problem you'll ever be handed. Memorize the 4 questions, not the answer. Now the wrong options ranked because that's where most of the points and most of the learning actually hide.
A. Simple diffusion. Tempting, because glucose is small and everyone pictures it just slipping in.
It's the main trap, and 2 separate clues execute it. Simple diffusion has no protein, so it cannot be stereoselective. It would carry D and L at the same speed, and the stem says it doesn't. And simple diffusion never saturates. There's nothing to run out of, so it can't plateau, and the stem plateaus. Either clue alone kills A, and underneath both, glucose is polar, so it barely crosses the oily membrane on its own in the first place.
D. Primary active transport.
A better trap because it is a carrier and it does saturate, so it sails through clues 1 and 2. The discriminator is the energy line. Primary active transport hydrolyzes ATP to pump its cargo against the gradient. The stem says flatly, no ATP, never uphill. So the very feature that defines a pump is the feature the experiment rules out. D dies on the energy line.
E. Sodium glucose cotransport. This one's a real glucose carrier, so it feels dangerous.
It's the strongest distractor, so give it real time. SGLT is genuinely carrier-mediated and genuinely stereoselective. It passes clues 1 and 2 just as well as the right answer. 2 things sink it. First, direction. SGLT moves glucose uphill, spending the sodium gradient to do it. And the stem says glucose here never goes against its gradient. Second, and just as decisive, location. SGLT lives in the gut lining and the kidney tubule, tissues that reclaim glucose against a gradient. These are fat cells. Adipocytes take glucose in, downhill, through GLUT4. They don't express SGLT. Wrong direction, wrong tissue.
And B. Receptor-mediated endocytosis.
The throwaway, 1 line. Endocytosis swallows big cargo. LDL particles transfer in whole proteins by wrapping them in a membrane vesicle and pulling them inside. You do not import a single small sugar molecule that way. It's like sending a shipping container to deliver 1 grain of rice. Recognize it and move on. Here's the highest yield 20 seconds. Because if you really understand the answer, you can predict how it changes. Change 1 variable at a time.
Go.
Version 1. Same fat cells, same experiment, but now glucose keeps piling into the cell even above its outside concentration, uphill, and the whole thing stops the instant you remove sodium from the buffer. 1 variable changed. It now runs uphill and depends on sodium. What's the answer?
That flips it to E. Sodium glucose cotransport. Uphill plus sodium dependent is SGLT.
And a bonus you'd now say without being asked?
These wouldn't really be fat cells anymore. That behavior belongs to gut or kidney. where SGLT lives.
Exactly, the mechanism drags the tissue with it. Version 2, subtler. Everything the same, but insulin does nothing to the plateau. Still stereoselective, still saturable, still no ATP, still downhill. Now what?
The mechanism is still C, facilitated diffusion, still a GLUT. It just isn't GLUT4 anymore, because GLUT4 is the insulin-sensitive one. Sounds more like GLUT1, a red blood cell, or the blood-brain barrier.
That's the one I most want you to take away. The mechanism, facilitated diffusion, and the specific protein, GLUT4, are two different layers. Insulin sensitivity is what picks GLUT4 out of the GLUT family. Drop it, and you drop down to a different GLUT. But the answer to which mechanism doesn't budge.
So the whole question in one breath. Stereoselective and saturable means a carrier is involved. Plain diffusion can't tell D from L, and never plateaus. Then ask two questions. Energy and direction. No ATP and downhill is facilitated diffusion. The GLUT family. Uphill powered by sodium is cotransport. The SGLT family in gut and kidney. ATP driven uphill is a pump, and the insulin raising the ceiling names GLUT4 in fat and muscle.
Read the kinetics, not the molecule. 4 questions: stereoselective, saturable, energy, direction, and every membrane transport question falls into place. That's the whole lesson.